We were talking about three different kinds of techniques, right? One is your mhm, okay. So, what are the three techniques we were talking about? One is viral mediated, then micro injection, and embryonic stem cells, right? So, viral mediated is like you are editing the viral genome such a way that you are introducing a foreign gene of your interest, then you are introducing the viral virus to transfect or infect the cells so that it will inject its genome into the cells. And of course it is a natural phenomena that the genome of the viral whole thing will be go and integrated into the genome of the host cell. So that host cell, what we are talking about here, most of the times it will be embryonic stage, different embryos like morula or the fertilized oocyte or the zygote or blastocyst stage cells. All this thing, any stage, whatever the thing you choose to take and infect those cells with the engineered virus such a way it will carry your gene of interest. Then if you allow the viral virus to go and infect, it will go and inject its genome and integrated the genome along with the foreign gene, that is transgene, into the host genome. So thus it will be keep on passing to the next cells. Whenever the cell divide, it will be going to be present. So if you are conducting that one at the earlier stage when it is a single cell, then all the cells are going to be resulted with having the transgene. That means whole uh, resulted animal will be carrying transgene. But if you are in doing the same thing at the later stages, then the problem is some of the cells may have, some of the cells may not have the transgene. So it may result into the mosaic animal. That mosaic animal means few tissues will be having the transgene, few tissues will not have the transgene. So if the few tissues which are having the transgene, by chance, if it is include the germ cell lines, so the next generation going to be fully transgenic animal. So that is what it works with viral mediated transgenic production. And let us see what happened. I mean, of course, the micro injection we have already covered. Maybe you can see once again. To obtain the transgenic mice by this route, females are super ovulated, mated to fertile males, and sacrificed the next day. Fertilized oocytes are recovered from excised ovule. So it's not so uh, no pleasant thing. That is why I I was not liking and I discontinued this transgenic animal thing. Of course, for the research, we should not think in that way, but you need to sacrifice a lot of rats and mice and all. So first, what you need to do, you need to make the um, you need to give a hormone to the female mouse that it will super ovulate. Otherwise, it will not have that many ovoms, right? So oocytes, right? So give a hormone such a way that it will get super ovulated. It releases a lot of oocytes. Then mate with the male, and then so it is expected to the having the fertilization. The next day need to be sacrifice that mouse, and then collect the oviduct and take out all the oocytes, fertilized oocytes from the oviduct. Then the DNA of interest is then micro injected using a micro manipulator into the male pronucleus of individual oocyte. So last class we have seen the how it will hold the fertilized oocytes and it injected. So the terminology wise, it is called as a micro manipulator, and which is releasing the DNA of your interest into the male pronucleus of the fertilized oocytes. Then surviving oocytes are re-implanted into oviducts of foster females and allowed to develop into mature animals. Okay, so this surviving oocytes, that means not many will survive because you are poking the cell, which is very delicate. Just cell membrane is there, so it's not so easy. And okay, whatever the oocytes, it's surviving, re-implanted into the oviducts of foster females. So as I told you in the last class, so again it need to be re-implanted into the uterus of the new different mouse. So that is a foster females we call as. And during this procedure, the micro-injected DNA randomly integrates into the chromosomal DNA. So just you are injecting into the pronucleus. That doesn't mean that it is go and integrate. So the next process is what? It is randomly go and integrate. So where it is going to integrate, we don't know. But it will go and integrate into the genome, usually at a single site. Although rarely two sites of the integration are found in a single animal. So most of the times you will expect a single site integration even though you can introduce many copies of the DNA. Individual insertion sites typically contains multiple copies of the transgenes integrated into chromosomal DNA as head-to-tail contamers, concatamers. Like it is not unusual to find 50 or more copies at a single set. So it is expected somewhere it is going to integrate, some break will be there in the DNA. So that it will it will make the allowing of this DNA to get integrated. So when it is get integrated, not one copy, it will go multiple copies, at least around five or more than that. It will get integrated. Same gene, okay, whichever you injected into the male pronucleus. So this is how you can visualize this. So the holding pipette with the vacuum, this cell, so gently, because it's a very delicate because the only cell membrane is there, and the micro injection pipette, so will inject the DNA of your interest into the male pronucleus, and then yeah, this is the one. After that it will form like a zygote, I mean fused nucleus of from the male and female. After that, what you are going to do, transfer into the oviducts of pseudo-pregnant female. So I told you what is meant by pseudo-pregnant in the last class. So the female will be mated with the fertile, sorry, infertile or sterile male, so that the uterus will be ready for the implantation. So in that stage, so this genetically engineered the fertilized oocytes will be injected into the uterus, so it will get implanted. So during this step, so you can see the how it will go and integrate. So once it get injected, so this DNA will be present in the nucleus, and this is the genomic DNA. If there is any possibility of having some nick chromosome, which usually happens, and it will get repaired continuously. But if this find any nick DNA, so it will go and integrate there. So it is random. So we don't know where it is get integrated. So then the that nicked place. So instead of one copy, is a multiple copies go and integrate. So that is what happens, and once it successfully done, then it is going to be a transgenic mouse. You will get the the generation, the founders. And the difficulties and disadvantages, as I told you, Um, One of the major disadvantage is, surviving rate is not so high. So that is one thing. And second thing is very expensive. The mission is a very big. And third one is very experienced and skilled professionals are needed, not like other techniques, just pipetting and incorporating. No, it is not going to work like this. So you need to work, you need to be have a skilled person to do this. So that is another disadvantage. And yeah, so another disadvantage, if you observe, either viral mediated or uh, micro-injected. So what is the major another disadvantage? We don't know where it get integrated into the genome. So if you don't know what happens, sometimes it may be integrate into the place where the very vital genes of the genome is present, of the cell is present. So very is vital. So if that it integrated in the middle of the any vital gene, what happens? So it may not, it will not survive because the vital gene is very important to be present. So that function is very important. So if it is not survive, that is also okay. But another problem is sometimes it will affect it will survive, but it will affect the normal metabolism if it is get integrated at the region where the other important genes are present. So that is another disadvantage. And one more disadvantage is if it get integrated somewhere um, in the genome where it is not get exposed to the RNA polymerase, like uh, heterochromatin regions. So where it is dense DNA chromosome will be there. So that place will not be exposed. I mean, it will not have any functional genes there. So just it will be a condensed chromosome. It's structurally present, but it is not having any transcribing genes there. So Joe, if you go and put there is like a someone released into a dark and a huge forest. So what happens is it cannot survive. So it cannot be transcribed. So that is the disadvantage. If you don't know where it get integrated. So that is why it is very important to integrate at a specific place. So you have to direct where it can be integrated. That is that will be a very good advantage. But using these two, it is not so clear we can direct it. So the next one which we commonly use is the engineered embryonic stem cell method. So this method we can actually direct where you want to get integrated into the genome, because you are doing in the petri plate. So the number, a lot of cells are there. So you can select which cell you want to use for the implantation into the mouse later on. So that is a very good advantage of embryonic stem cell. So let me explain to you how it works. So the micro injection of the foreign DNA into fertilized oocytes is a technically difficult and not suited to large scale production of transgenic animals or to sophisticated genetic manipulation. So as I told you. And a popular alternative, but one which is so far restricted to the construction genetically modified mice, involves transferring foreign DNA initially into cultured embryonic stem cell. So how this embryonic stem cell culture you can initiate? You have to take embryos from the mouse, and which is on the blastocyst stage. So as I told you, I showed you there is a single layer outside, and inside will be a cell mass, and a big space also will be there. So the cell mass, each cell is a totipotent. That means totipotent stem cells are the stem cells that can be developed into any type of specialized cell of the body. So we have hundreds of different cells, different types of cells in our body. So this totipotent stem cell can able to differentiate into any kind of cell of the adult mouse. So that is what we call as totipotent. Any type, all the kinds of cells. So that is the totipotent. So the desired gene is inserted into this cell. So take those cells and put into the petri plate and culture it, and you inject, not inject, there are some methods how you can introduce the foreign genes, and you just make this one to the desired gene can be inserted into the cells. The cells contain the desired DNA or incorporated into the host embryo, resulted into chimeric animal. So after, there is some methodology, you have to introduce a foreign gene of your interest into the cells, and there is a methodology to screen whether it is get integrated or not. So once you satisfy according to your need, it is get integrated into the genome, those cells you take and put into the new animal, new female, a new foster female. So what happens though, that is going to be resulted into chimeric. So usually that one will be introduced into a different blastocyst instead of directly introducing into the uterus. So usually these embryonic stem cells first will be introduced into the a different blastocyst, and then these blastocysts will be introduced to the new embryos. So this is how this visualization for you. Um, See, this is the embryonic stem cells growing into the tissue culture. So from where it comes, these embryonic stem cells, see this blastocyst, like otherwise you see this blastocyst. So where it will be having a single layer of cells outside and inside the cell mass. So these all inner cell mass are called as totipotent. So take these cells and put into a petri plate. So actually the different animal, just to explain to you what is how it looks like the blastocyst. I'm just explaining here, but so it has been taken and keep growing in the laboratory for longer period. It will survive for longer generations without any issue. That is the property of the stem cell. Stem cell is the mainly the uh, property is one property is the stem cell can divide and give the stem cell. Okay, and second property is it can go many cycles. So keep going, the same property will be given to the next cell. And what is a property? It can able to differentiate at any time, given a signal, a different specific signal. The signal can be anything, some chemical, some temperature, some attachment, or some physical attachment, anything, and pH, anything, some different different signals needed for uh, different specification. So this stem cell can able to differentiate it into a different kind of cells. Okay, so if the stem cell need to differentiate into narrow cell, maybe it needs some signal, some chemical. That signal can be anything, either chemical or some kind of a protein or some kind of a physical interaction, different. Okay, that is just collectively you just understand like a signal. Okay, so what I said, these totipotent stem cells are able to differentiate into any kind of cell of the adult animal, and also it can able to generate the stem cells in the by dividing. So you keep doing this culture in the laboratory. That is what we call as stem cell culturing. stem cell laboratory. Okay, so this how the you will see the petri plates and culturing. It's not so easy task. Huh? Stem cells growing is not like normal cell lines. It's very difficult to maintain. Um, very difficult. Okay, so this foreign gene you just introduce into these cell lines, cell culture when it was a stem cell. So randomly, when there are different methodologies like uh, that is called as a transfection. Different methods like again here also virus mediated or chemical mediated or the the liposome mediated or nanosome mediated. There are different methodologies are there, so which you will learn in the next semester. There is a course called as cell culture, or if it is a new batch of students, it is incorporated into the animal biotechnology. So any of this course, you will understand the methodology of introducing a foreign gene into the cell culture. So it will be a stem cell or normal eukaryotic cell lines. Any cell lines, how to introduce. There are so few methodologies are there so that you will understand that. So basically, you just imagine there is a one of the method is viral mediated. Just know what I explained to you. So take a virus and manipulate or integrate the gene of your interest into the viral genome, and those virus you infect these cells. So what happens? These cells, the virus will be injecting its DNA into the these stem cells, and it will go and integrate it into the gene, its genome. So that is viral. And the liposomes is like is a lipid bilayer, and and the DNA will be present inside you. It's a very simple, the liposomes are available commercially. Just you mix with the DNA together, it will form liposome DNA mixture, and you just introduce. There are some kind of specific mediums you need to add during the time, and the cells will get incorporated inside this the genome. So like that, there are different methods. So just to make you a little bit imagine, so I just give you some example. So the detailed methodology you will understand in the cell culture. So whatever it is, so there are some transfection methods. This is called as transfection methods. So different transfection methods allows to introduce the foreign gene of interest into the stem cells. Okay, so randomly some of the cells only will go. So efficiency is not that high. Usually 1 in 100 or something only get the foreign gene. So 90 to 99% of the cases, it will not get any incorporation of the foreign gene. So here the see the you think as a disadvantage that only the 1 in 100 or something, but that is having the similar same time we have uh, the tools, genetic engineering tools, such a way that we can screen which cell is having incorporated the gene of interest. So that particular cell, you can collect it and grow into a different petroplate. Only the transgene containing cells will be grown. So what is that method? So the, the selection method. So one of the way you understood in the bacteria, right? So what is that selective method? You can guess what it could be. Hello, are you following what I'm talking?
Yes, sir.
So can you say what could be one of the selective method to take the cells, select the cells which are having the transgene? Very good. Vivian and Tan A. Kang. So tr- just introduce the antibiotic gene. But this antibiotic should be different because prokaryotic antibiotics only restrict the or kill the bacteria. Here, eukaryotic antibiotic you need to take. So, like neomycin kind of thing. So, where it will affect the eukaryotic cells. So, when you are injecting this gene, the orange color gene, so it's not only your gene, you put along with your gene your antibiotic resistant gene to the neomycin. So then what happens, you just inject, I mean, there are different ways you can introduce, and the cells whichever it got incorporated, so definitely you will not see like what you have seen in this in the slide, the orange color or something colors you cannot see. But what how you can select to grow only those cells which is having your gene of interest. So you grow these cells after the transfection, you grow the cells in a medium which is containing neomycin antibiotic. So once it is having, if you it is having a neomycin, so the cells which are not having the neomycin resistant gene will not survive. Just it will die. So only the cells which is having the neomycin will be present. Okay. So those cells. So what is the next step? You just keep growing into the in the laboratory. Of course, these cells you can also preserve some of the cells. You can preserve cryopreservation. There is another technique you will learn in the cell culture. So using the cryopreservation, you can some of the cells you can preserve it, and some you can introduce into the like yeah, here again the mouse and different mouse you have to take, mate with a male, and after 3 days take the embryo, that is a blastocyst stage. In this stage, you collect this blastocyst and introduce the engineered embryonic stem cells into this embryo, into the cell mass. So what happens? It will be like this. It will occur like this: early embryo partially form the embryonic stem cells, and then this you are going to introduce into the different female again. So introduce early embryo into the pseudo pregnant, again is a pseudo pregnant. So I told you how it need to be produced pseudo pregnant. So this blastocyst will be introduced into the pseudo pregnant uterus. Then it will give different animals, so different mouse. So randomly these cells, so engineered stem cells can lead to the any kind of cells of the body, not all the cells of the body, because different other cells also there. So all these cells go and differentiated into different types of the cells of the body, and different tissues of the body resulted into different possibilities. So whatever the possibilities, they are all a kind of chimeric, because only part of the tissues, part of the body only having the transgene, so not whole animal. Okay. So if that random differentiation leads to the criteria where this transgene is present in the germ cell lines, okay, if that have tend to have the germ cell lines having the transgene, the next generation going to be fully transgenic. If that is germ cell line is not having something like this, just part of the the upper part of the body is having the transgene, but not the germ cell lines not having the transgene, so that will not lead to the next generation transgenic mice. So next generation will have normal animal, not even chimeric, just normal mouse you will get. So if the chimeric mouse which is carrying the germ cell line transgene, then it will lead to the next generation transgenic mouse with one copy of transgene replaced with the alternative gene in germ cell line. Okay, so that is the thing. So now another important thing we need to uh, sorry. Okay. So, for example, the desired gene can be ligated to a marker gene, such as a neomycin resistant gene, enabling a positive selection for cells that have been successfully transfected. So those kind of things, which is not possible just viral mediated, viral mediated incorporation directly to the embryonic embryo cells. So you cannot select it. Okay. And also in case of a direct micro injection, so just you need to do the micro injection and just you need to put it back for the implantation. But in case of embryonic stem cells, as it can keep growing inside in the laboratory on the petri plates, you can do whatever you want and you can screen until the desired thing obtained. For example, you can incorporate your transgene along with the neomycin resistant gene along with that and introduce that using the different transfection methods, and then you can select the cells which can able to grow in presence of neomycin resistant neomycin antibiotic. That is called as positive selection. So that means the wherever the gene of your interest present, that cells will grow. The remaining cells will not grow. That is called as positive selection. The presence of desired gene can be verified quickly by a PCR-based assay. Embryonic stem cells also offer a huge advantage of gene targeting by homologous recombination, a method which can permit a programmed selection alternative of a single predetermined gene and also highly specific way of chromosome engineering. Such approaches are extremely powerful for the understanding of the gene and also creating um, animal models of disease. So the idea is, if you want to incorporate your gene of interest at a specific place, how to direct it? So using the homologous regions. Homologous regions are the regions like exactly similar to the animal's genome, where the some locus you take, that locus sequence, if you take exactly similar to the locus sequence, if you take adjacent or flanking regions of your transgene, and there is a chance that these homologous regions and when you introduce this DNA to the cells, go and recombine. That is called as homologous recombination. I will explain to you. So thus you can direct where it if you want to go and integrate the transgene into the genome. So those things you can do. So we will learn now methods for detecting the correct gene integration. That means you want to integrate the transgene into the genome at a specific location. So how to do that? So that we will learn in now, and there are two different methods I'm going to teach you. One is positive negative selection. Another one is PCR selection. Okay. First, let us understand what is a positive negative selection, and then we will move to the PCR selection. Okay, so let me repeat for you. You have a gene of interest which you wanted to integrate into the cell, which is called as a transgene, and we want to incorporate into the cells somewhere in a specific chromosome and specific site of the chromosome. In that place, you need to select first your the sequence where you want to incorporate that. You name that one as homologous block 1 and block 2. So that regions you have to take the same sequence adjacently to the transgene. So this is going to be your full construct of your DNA, uh, where you are incorporating or introducing you into the stem cells using different transfection methods. So this is we call as homologous block 1, homologous block 2. And this one you are introducing into the stem cells using different transfection methods. Okay, this is called as transfection. So once it get introduced, transfected, so this transgene will incorporate into the stem cell genome. There are two ways. One, it will just go and integrate into the any of any place of the chromosome and any chromosome. That is one thing. That is called as a random integration. So with this, we don't know where it is get integrated and which we don't want that, isn't it? And the second possibility is this homologous block 1 and block 2. So it will go and put there, bind each other, and your transgene. It will get homologous recombine. So it's like this. Your genome will be there. So very big genome, chromosome, and where you will have a homologous block 1 and homologous block 2. You just name it only. This is nothing but is a sequence. Okay, but you are taking the same sequence here and there. So we just call that one as homologous block. So when it's go and find its similar sequence, so it may go for recombination. Just recombine each other. So it's go like this and take this and like this. So thus, once it gets recombined, what happens? The transgene going to be come here, and genome sequence is going to be go here. Okay, and this is the transgene. So this process we call as a homologous recombination. So that is what actually we want, because that will direct where you want to have your transgene. So that is what the desired one. That without affecting, there are so many reasons. Without affecting other genes, another thing is that gene, that place is required to express and produce the product in your desired way. Maybe it is having a promoter which can be, it can affect the transgene production in such a way that in which way you want to have. So there are so many reasons you want to have your transgene at a proper location, desired location. Okay, the desired location is possible by introducing the homologous blocks where you want to introduce your transgene and introduce the in your construct the DNA piece which you are transfecting into the stem cells. So adjacently you put homologous blocks to the transgene, so that once you transfect this whole DNA and once it go into the nucleus, it finds the homologous regions, and then it is go for the homologous recombination. Then does the transgene get incorporated into the genome at the desired location, at the planned location. So that is the second possible way. Okay. So second one is incorporation at desired location. So what is the third possibility? The third possibility is no integration. So no integration also possible, right? There are all three possible. So random integration, integration at a desired location, or no integration. Because I told you, once you introduce, it doesn't mean that all the cells will take it. you are introducing into the medium only. So all the cells are there. So there are different transfection methods and different efficiency. It may lead to the some percentage, maybe 1% or 5%, whatever the percentage of the possibility it go inside, and what is the possibility go to the nucleus and integrated. So there are, you most of the times you end up with the majority cells no integration. So only very few cells get integrated. There is also two types: random integration and desired location integration. So what we want is only integration at desired location. So how you can screen those things, how you can select the cells which is having at only integration at a desired location. That is our important thing. So that is what we are seeing, the positive negative selection. So in that, what usually you have the construct. The construct is a, I mean, just usually we are constructing pieces of DNA genetically engineered such a way that that piece of DNA you are introducing to the stem cells so that transgene will get incorporated. So usually we call this one as a construct. Okay, so transgene. You will put one more gene here, which is neo-R, abbreviated as neo-R. I think you can guess that is having it will code for a neomycin resistant gene. That is the enzyme which can degrade the neomycin. So just like ampicillin resistant gene or kanamycin resistant gene, here neomycin resistant gene will be there. So after that, you will have the homologous blocks. These are all DNA sequences only, okay? Just to make you clear, understand just by putting in boxes. Otherwise, just a DNA sequences only. Okay, so what is this? Homologous block 1, homologous block 2. So apart from that, you are going to have two more sequence. Here, you will have another gene we call as TK1. And here you will have another one, TK2. So these two genes are the genes which will produce a protein called thymidine kinase enzyme. Thymidine kinase. So these are the actually taken from different virus. So, but the purpose of that existing was different. But as I told you, many of the genes in genetic engineering we exploited or using for our genetic engineering, efficient genetic engineering purpose. So this thymidine kinase 1 and 2 from the different virus we have taken and put here. So I told you, I will tell you what is the need of this thymidine kinase 1 and 2. And I think roughly you have already understood what is the use of the neomycin resistant acid, right? So now we will see, again, we will see the three possibilities. One possibility, first one is, let's see the no integration, the third possible, there is no integration, right? So you will have the cells inside the Petri plates, the stem cells. So the one which is the many of the cells will not have any integration, right? So you introduce this one to the cells, and the third possibility, there is no integration. So what happens when there is no integration? So still the cells will be growing in the normal medium, but you add neomycin to the medium. What happens? The cells which is not having this any construct, will die. So maybe majority will die. Okay. So a few will be there. So you already excluded these possibilities, right? So this is called as positive selection after adding the neomycin. So because the cells uh, which are having the neomycin resistant cassette will survive. The cells which are not having the neomycin cassette will not survive. That is what when it is having the gene will survive, when it's not having the gene will not survive. So that is called as a positive. When the something is present, it will survive. Something is not present, it is not survive. It's not only the neomycin. So what I'm trying to say, positive, why it can named as when the some gene is present, the cells will survive. When that particular gene is not there, it will not survive. That is called as positive selection. Here, the neomycin resistant cassette, if it is present, this will survive. Neomycin cassette is not present, it will not survive. It's clear, right? So that is how you can remove the possibility of no integration possibility. So we have the two other possibilities. One is a random integration, another one is homologous integration. So random integration, what happens? So when it goes for the random integration, uh, let's imagine this is the genome, and there is no any homologous blocks there, and in that, due to the some nicked gene, nicked area, the whole thing go and integrated over there. So what it will result? What it will result? It will result into like this, right? So genomic DNA will be there, and you will have the thymidine kinase 1, then homologous block 1 and homologous block 2, then transgene, then neomycin resistant cassette. Also, you will have thymidine kinase 2. Everything will be there. Everything will be there along with the, yeah, everything will be there, and the whole genome. The big genome will be there, chromosome, but it will have your gene of interest along with the everything will be there. Okay, so when it will be there, when all will be present, if it undergone just random integration at any nick DNA. So other possibility is what? Other possibility is what? So it is get integrated at at a desired location. That means it will just use the homologous recombination. When it go for the homologous recombination, it will have, because these 2 points are important. So these 2 points get integrated when homologous recombined. This region between the homologous blocks region only get incorporated here. The outside one will not get incorporated. That is the main thing you need to understand here. So homologous block 1, homologous block 2, in between region only it will get incorporated into uh, the desired location. So the remaining thing will not get incorporated. So that means thymidine kinase 1 and 2 will be not get incorporated. That is what I'm trying to explain to you. So there were three possibilities I told you. One is no integration. No integration is already cleared off because nothing will be present. None of the genes will be get incorporated into the cells. So in presence of neomycin, so already the cells die because neomycin resistant cassette is not present. Now We will see the first two scenarios. One, there is a random integration. Random integration leads whole thing going to be incorporated, including the thymidine kinase and thymidine kinase 2. And here, in case of desired integration, that is due to the homologous recombination, only homologous block 1 and 2 and neomycin resistant cassette and transgene, only this will be there. So there is no thymidine kinase. So can you able to see the difference between the random integration and the homologous integration? What is missing? What is extra present in compared to the both? Can you able to see, right?
Yes, sir.
Okay. So thymidine kinase 1 and 2 are present in case of random integration. Thymidine kinase 1 and 2 will be not present in case of desired location integration due to the homologous recombination. So now the thing is how to get rid of this one. So now you can understand what is a function of the thymidine kinase. Thymidine kinase is a gene will produce a thymidine kinase an enzyme which will act upon a substrate called gan-cyclovir. Gan-cyclovir is a substrate, and it will produce a product which is a poison to the cell, which is a toxic to the cell. Again, I repeat, thymidine kinase gene produces thymidine kinase enzyme, which will act upon a substrate called gan-cyclovir, and this gan-cyclovir will be converted into a toxic compound upon the action of the enzyme thymidine kinase. So see how it will be utilized here. So now the cells in the Petri plate, normal medium will be there, medium plus neomycin and plus gan-cyclovir. Since gan-cyclovir, what happens when you add this gan-cyclovir? If the cells are having a random integration, the thymidine kinase genes will be there. This thymidine kinase will act on the gan-cyclovir inside the cell. Once gan-cyclovir present in the medium, of course, it will enter into the cell, and that gan-cyclovir will be converted into toxic product, and cell will die. The cell will die. When cell will die? When the thymidine kinase is present. So the cells which are having the just neomycin resistant cassette and transgene, which are not having thymidine kinase. So in presence of gan-cyclovir, the gan-cyclovir is not converted into the toxic compound, and the cells will not die. So what you understand now? The cells which are having the thymidine kinase will die, which are not having will survive. So the same way, similarly, what you can understand, the cells which are something is having, some gene is having will die. Something is having, it will die. Something not having, it will survive. That means it's a negative. Previously what? Neomycin resistant having, it will survive. Here, something having, it will die. So this is a negative selection. So thymidine kinase. So the thymidine kinase, whichever the thymidine kinase genes are present, it will die because it will act on the substrate called a gan-cyclovir, which you are going to introduce into the medium. After the transfection, what is the thing you need to do? You need to add two extra the components to the medium. One is the neomycin, another one is gan-cyclovir. So automatically the neomycin will eradicate the cells which are not having any integration, and the gan-cyclovir makes the cells to die wherever the thymidine kinase incorporation. Thymidine kinase incorporation it was resulted due to the random integration only. When it is incorporated due to the homologous recombination, only it will incorporate only this region. This region will not get incorporated. So it will survive only the cells which is having homologous recombination. That is what we want. Homologous region we have taken such a way that where you want to get incorporate the gene of interest. So the finally by using this screening techniques, you will direct and you can select the cells which are having your transgene gene of interest at a desired location. Those cells you can take, you can culture to get more cells into the culture medium, and then you can incorporate into the new blastocyst, and then you can implant into the uterus of the foster female, and then it will lead to uh, the chimeric or mosaic animal. And then you need to select whether you need to screen different techniques, like whether the transgene is present in the F1 founder animal or not. Then you have to go for the second generation. You may not get immediately the transgene in any small tissue if you have taken, because some of the tissues may not have the transgene. So usually you have to wait another generation. So go for that second generation. Then second generation, if it is having, that mean the founder animal is having the transgene at the germ cell line. So then second generation, if you have the transgene, that means that whole animal will be having the transgene at a desired location. So this we call as a positive negative selection. Let me share the screen. Yeah. So after transfection to the embryonic stem cells in the culture, the DNA vector is designed into to integrate within the specific chromosomal location. Some cells have DNA integrated at non-target sites, DNA integrated at the correct sites, and no integration at all. So all possibilities there. To enrich the DNA integration at the correct site, the procedure called positive negative selection is used, and this strategy uses the positive selection of the cells that have vector DNA integrated anywhere in their genomes, and a negative selection against the any vector DNA sequence that is integrated at non-target sites. So the targeting DNA vector for positive selection procedure, usually the two blocks of DNA sequence that are homologous to the separate regions of the target site. So homologous block 1 and homologous block 2. So neomycin resistant cassette and transgene. These two must be present in between the homologous block 1 and 2, neomycin cassette and neomycin gene and transgene. So thymidine kinase 1 and 2 will be outside, must be outside of the homologous blocks. A DNA sequence coding for the neomycin resistant gene, and two genes which are thymidine kinases from HSV1 and HSV2. This is the virus we have taken. And then the arrangement of this sequence is a key. So arrangement, I just told you, the transgene and neomycin must be between the homologous block 1 and 2. Maybe these two can swap, not an issue. So the first transgene and the second neomycin, or first neomycin and transgene, that doesn't matter, but the both should be between the homologous block 1 and 2, and the thymidine kinase 1 and 2 must be outside the homologous blocks. And this is what the one I just told, that random integration, which is also called as non-specific integration, so will leads to the integration of the whole construct. That means including the thymidine kinases along with the homologous blocks, the neomycin resistant cassette and the transgene. Of course, neomycin resistant cassette is there. in presence of neomycin, it will survive. But uh, yeah, this is the one which is the one resulted due to the homologous recombination and the selective site. So homologous recombination will occur and it will lead to this one. So only neomycin resistant cassette and the transgene will get incorporated. And the positive negative selection, a result of non-specific integration, both genes for thymidine kinase 1 and 2 and the two DNA sequence that are homologous to the specific chromosome region in the recipient cells, homologous block 1 and 2, a gene neomycin resistant cassette that confers resistant to the cytotoxic compound G418 and the transgene and and are incorporated into the chromosome. After transfection, cells are selected for the resistant to the both G1418 and the compound ganciclovir. So this is the neomycin, okay, G147 and a ganciclovir. So which means cytotoxic to the cells that synthesize the thymidine kinase. Other non-homologous integration may occur and produce inserts with one or more other of thymidine kinase genes. After treatment with G1418 and ganciclovir, all the cells with a non-specific integration of the input DNA that includes the least one of the thymidine kinase genes are killed. So result of the homologous recombination, yeah, this is the homologous recombination which is not having thymidine kinase 1 and 2, so there will be no issues. Okay, so the similar thing, and here you can understand very clearly. When the transfected cells grown in the presence of neomycin, all cells lacking neomycin resistant gene will be killed. That is the positive selection. When the cells grown in presence of ganciclovir, gan, um, ganciclovir, so maybe some spelling mistake. Okay, ganciclovir, um, as well the cells that express thymidine kinase will be killed because thymidine kinase converts ganciclovir into toxic compound that kills the cells. That is called as negative selection. The cells that survive this dual selection scheme will have dual integration at target site. So that is about the positive negative selection. So then the same thing, how whether the cells got specific or directed integration or not, we can also screen by PCR selection.